Today we're going to be talking about the cell membrane. So far we've talked a little bit about molecules and what molecules can do in order to combine to make other molecules, and we've talked a little bit about the different types of cells. We're going to journey through the cell in order to see how the cell carries out its daily activities. And in order to get into the cell, we need to pass through the cell membrane. The cell membrane has a couple of names that it's often referred to as. Not only the cell membrane, but sometimes they also refer to it as the plasma membrane, and another term, which we'll explain in a minute, the lipid bilayer. We've already learned the term lipids in the last unit, where we were talking a little bit about organic compounds. Remember, those were our fats. The cell membrane has a couple of jobs. One of the jobs that the cell membrane has is to separate the inside of the cell from the outside environment. It helps to kind of contain everything inside, all of those organelles, and separate it from its surroundings. If we were to zoom in on a specific section of that cell membrane or that plasma membrane, it might look like this if we were to look at it from a side view. You can see it has several components. If this were the inside of the cell and this were the outside of the cell, you can see it's actually made up of two of these layers of what we're going to call lipids and fats, and that's where we get the name lipid bilayer from. It's actually two floating layers of lipids on top of each other. You can also see that there's this green section in here, which isn't actually green in the cell, but for this purpose we're going to color it green to separate it out so you can see the difference between this particular item called a protein. This is actually a channel inside the membrane made up of proteins, and we also have some carbohydrate markers which basically help to identify the cell as what kind of cell it actually is. So this is our somewhat microscopic view of the cell membrane, and if you were to imagine this going all the way around the cell, it almost serves as somewhat of a protective layer. So let's look at some of the other jobs of the cell membrane. The cell membrane helps to regulate or control what enters and leaves the cell. It obviously can't separate it from the entire outside environment. Sometimes bad things do get into the cell. We do call it selectively permeable, though, or semi-permeable. And if you think of what the word selectively actually means, it means to choose. Some things get through, some things do not. Permeable will mean to move or get through. In terms of what things can actually get through the cell membrane easily, small, non-charged, like a plus or a minus charge molecules can easily move into and out of the cell membrane to get into and out of the cell. Some of those things would include like your small gases like oxygen and carbon dioxide, and even something like glucose, which has a special transport channel. Oxygen and glucose would actually move in between these little lipids in the bilayer and squeeze in between the spaces because they're such small molecules. There are some molecules, though, that do not move through the lipid bilayer easily. Usually it's the larger or charged molecules that have a plus or a minus charge to them, which we'll often refer to as ions. Some examples of these things: starch is a very large molecule that cannot fit through, sodium, and chlorine. The salts, which we typically call those, are ions, cannot fit through this lipid bilayer easily and pass through. So how do things actually really move through that plasma membrane or cell membrane? Well, if we look at the membrane in a very simplistic view, we could consider the inside and the outside of the cell, and then this little layer in between that has these tiny, tiny little pores in between those lipids that would allow things to pass in and out. Now, in terms of the way things travel into and out of the cell, we refer to the concentration. You may have learned concentration back in the middle school, and it's called the amount of solute in an area. And if you can remember, solute is something that gets dissolved into another substance. So typically we'll talk about things that are dissolved in water as our solutes. In terms of concentration, we usually refer to high concentration and low concentration. High concentration would mean that there's more solute, or we dissolved more things into our solution, and a low concentration would mean that it has less solute or fewer things dissolved in that particular solution or water. These are really only comparison words, though. We don't necessarily have specific values in this particular case. It's relative to each other. So comparing the inside to the outside, we might say it's higher or lower. In that case, sometimes we talk about a concentration gradient, which is really just the difference in concentration between two areas. So we may go from high to low concentration. That would be called with the gradient. And against the gradient would be considered going from low to high concentration. Sometimes I like to think of the gradient going with the gradient, high to low, almost like a waterfall, going across and down the waterfall from high to low. I'd like you to pause and try, after we discuss this for a moment, in terms of labeling which side is a high concentration and which side of this membrane has a low concentration. Remember, this particular dashed area represents the cell membrane and the pores in between, and the purple dots in this case are going to represent our solutes, or the things that got dissolved in the solution. So I'd like you to label one side with an H and one side with an L, depending on which side you think has a high or a low concentration. So why don't you pause and try and see how you do. So let's see how we did. This side I labeled with an H. The reason why is because if you count the number of purple dots here, 1, 2, 3, 4, 5, 6, 7, 8, 9, there are more dots on this side compared to this side, which only has 4. So there are more solutes, or things dissolved in that given area, compared to the other side that has less. When we talk about movement through the channel or movement through the membrane, remember we're going in between this little lipid bilayer. And the term that we're going to refer to this as is called diffusion. It's a very big term for us. And the definition of diffusion is the movement of molecules from an area of high concentration to an area of low concentration. And one very key thing is that this movement does not require energy. And energy in our cells is referred to as ATP. The area from high to low concentration, remember now, is diffusion. So if we were to look at this particular model again that we already labeled high and we already labeled low, I'd like you to pause and try for a moment and try to decide which side you think the molecules would move towards using diffusion in order to have that movement. Which way would they go? Would they go from this side to that one, or that side to this one by diffusion? So let's pause and try and see how you do. In the case of this model, if I'm moving from high to low concentration, my molecules would actually move from the right side to the left side until it reaches something that's what we call an equilibrium. An equilibrium is almost what the word actually sounds like, equal. The molecules are going to move from one side to the next, from high to low concentration, until they basically equal themselves out. And then any further movement, it will be basically one molecule for one molecule. One might come over here, then another one has to go back. It'll maintain that what's called a dynamic equilibrium. It might change a little bit. One might go in, one might go out. 3 might go in, and 3 may come out. But essentially it wants to move towards what we call an equilibrium or a balance. Let's take an example from the human body. In terms of the human body, inside of our lungs we have these very small sacs called alveoli. And the alveoli is where the gas exchange occurs in order to let oxygen from the air actually go into our body to be used in order to make energy for us for chemical reactions. The way that things are actually going to move is we're going to bring the air into our lungs, into these little sacs called the alveoli, and somehow these molecules need to get into our bloodstream to travel to the cells. And the way that they're going to do that is based on diffusion. So I want you to think for a minute, what is in the air that we breathe? Is there more oxygen or is there more carbon dioxide? If you said oxygen, you did a great job. As we breathe in and take a breath of fresh air in, there would be more oxygen in that air than there would be carbon dioxide. So what I want you to do is I want you to pause and try, and I want you to think about which direction the molecules would move in order to get the oxygen into our body and for our body to get the carbon dioxide out back into the air around us. So pause and try and see if you can draw an arrow to show which direction they would move. Let's see how we did. So this is a blown up view of the drawing that we had on the other board of the alveoli. And if you think about the air that you breathe into your lungs, fresh air, that fresh air has a very high concentration of oxygen compared to carbon dioxide. So if we remember that definition from before, that the molecules are going to move from an area of high to an area of low concentration, we also need to think about the bloodstream. The blood is going to be traveling from the body to the lungs. And along the way, it's going to pick up a waste product, carbon dioxide, as we carry out our life processes. It will have used the oxygen in order to create energy for our cells. So in terms of the capillaries, as the blood travels along these alveoli from the body cells, they would have a low concentration of oxygen and a high concentration of carbon dioxide. Inside alveoli, remember, they're going to have high oxygen and low carbon dioxide levels. So in this case, our oxygen is going to diffuse from an area of high concentration to an area of low concentration, from the alveoli into the capillary, the bloodstream. And the exact opposite would happen for carbon dioxide. There's more carbon dioxide in the blood, so it would diffuse from the bloodstream, where there's more, higher concentration, into the alveoli, where there's an area of lesser concentration of carbon dioxide. If we were to look at this in a blown up view, here's our plasma membrane. If this were the side from the lungs and this were the side from the capillary, the oxygen would be going from the lungs into the capillary, carrying our blood, and the carbon dioxide would be traveling from the blood and the capillaries into the lungs, squeezing in between these molecules of the lipid bilayer. Now we do have a question. What about some of the larger molecules that are really important for life? If you remember back to when we were talking about some of the organic compounds, one of the organic compounds that we really knew was very important was glucose. Glucose is a slightly larger molecule than something like oxygen or carbon dioxide, but our cells need it tremendously for life processes. So one of the things that can happen is something called facilitated diffusion. Facilitate means to help. So in this case, there's some sort of helping going on with diffusion. And the definition is the movement of molecules through membrane channels. It's still called diffusion though. So remember, it's going to go from high to low concentration. There will be no energy or ATP needed for this process. But since glucose is such an important molecule, we're going to use these special protein channels in order to help glucose move into and out of the cell through the cell membrane. It's just a little too big to fit through these pores in between the lipid bilayer though. So it's going to move from an area of high concentration to an area of low concentration using those special protein channels. Since it's still diffusion, there won't be energy needed. We're going to talk about how even larger molecules get through the membrane at a later point. For now, we're just going to stick with diffusion. No energy needed, no ATP, and we're going to move from an area of high or greater concentration to an area of low concentration.